Hey guys, so in this video we're going to be going over lab exercise 7, which is about photosynthesis and respiration. So what you need to know is that our plants, they can both photosynthesize and respire. During the daytime, they're mainly using photosynthesis, and at night they switch to relying on cellular respiration. So when we're looking at the figure, what we see is that in the daytime, when we are photosynthesizing, the plant is going to take in water and it's going to take in carbon dioxide. As a result, it's going to produce glucose and release oxygen, just like this photosynthesis equation shows. So here we have carbon dioxide being consumed, water being consumed, and sunlight. And then as a result, we're releasing oxygen and glucose. Then at night, when we have no light, cellular respiration is going to take over. And cellular respiration, it's going to use oxygen, and it's going to consume glucose, which is exactly what we're seeing here. And it's going to produce carbon dioxide and release water and energy in the form of ATP. So something really important to understand is that even though these processes seem like they are distinct from each other, seem like they are happening separately, they actually do occur or can occur at the same time, especially when that plant is exposed to light. But they don't happen in the same place. Photosynthesis is going to take place in the chloroplasts, and cellular respiration is going to occur in the mitochondria. So the reason that photosynthesis and cellular respiration can occur at the same time is because they serve different purposes. Photosynthesis is only the only one that is going to require sunlight. So the purpose of photosynthesis is to capture light energy and create glucose. Meanwhile, cellular respiration is going to break down glucose to produce ATP, which is the energy that the plant uses to power its cells. So even while that plant is making food through photosynthesis, it still needs energy to grow, to transport materials, and to function. And that energy, it comes from cellular respiration. So that's why both of these processes can and do happen at the same time. So the purpose of this lab, it was to study how carbon dioxide is produced and consumed during both of these metabolic processes. It was to compare where starch is being stored in a leaf with where chlorophyll is found, and then use paper chromatography to separate different photosynthetic pigments and to analyze the absorption spectrum of one or more pigments. So during this experiment, what we were measuring was gas consumed. Right, so we were measuring gas in the form of carbon dioxide. So I'm sorry, not gas consumed, the release of carbon dioxide, which is a gas. So we were measuring the release of carbon dioxide. So it's really important to remember that during photosynthesis, we consume carbon dioxide, and cellular respiration produces carbon dioxide. So knowing these basic formulas, that is why it is key to understanding the data that we are going to obtain. So to set up the experiment, what we did was we took a light source. Let's see if I can draw a lamp. We took a light source, right? So the lamps were something like this. And what we did was take a beaker. So we took a beaker that looks something like this, and we filled it with water. And behind this beaker that was filled with water, this is where we put our plants, leaves, inside of a little container that had our sensor. The reason for this beaker with water, it was to act like a heat shield for the leaves. Basically, it was just going to protect the leaves from the heat of the lamp, and it's just to help keep that environment stable. So at specific moments, we covered the plant with foil and turned off the light. What we noticed was that carbon dioxide production increased when the lights were off. And just look here. When the lights were off, we had carbon dioxide going up, and when the light was on, carbon dioxide was going down. When the light was off, it went up, and when the light was on, it went down. So this makes total sense because in the dark, photosynthesis stops and the plant is only using cellular respiration to produce carbon dioxide. So if carbon dioxide is going up, then oxygen has to be going down since it's being used during cellular respiration. On the other hand, when the plant was exposed to light, we saw that our carbon dioxide levels decreased because now photosynthesis was happening and it was using that carbon dioxide. So what that means was that oxygen was also increasing because oxygen is a byproduct of photosynthesis. So what we can say is that if carbon dioxide decreased, photosynthesis occurred. If carbon dioxide increased, we had cellular respiration. But we can also determine that if our graph was flat, then neither of these metabolic processes is going to be dominating. So in part B of the lab, what we did was we looked at that relationship between photosynthesis and starch production. We know that our plants, they use chlorophyll to carry out photosynthesis, and photosynthesis produces sugars. We know that starch is a sugar, and we know that IKI is a test that is going to detect starch. So to figure out where photosynthesis had occurred, we used IKI. So we first started with a coleus leaf. And this coleus leaf, it has different pigments. And the green areas, they have chlorophyll. The red or purple areas, they have anthocyanins, right? So it's anthocyanins. Green is chlorophyll. And wherever we have this white or off-white color, what we have is no pigment. So to run the test, the first thing that we did was put that coleus leaf in boiling water, which removed the red pigment. These red pigments, they are water soluble. So they disappeared in water. Then we put the leaf in alcohol in a hot water bath, which removed the green pigment. That means they are alcohol soluble. So chlorophyll is alcohol soluble. So once we had this decolorized leaf, we then applied IKI. And anywhere that the IKI turned dark purple or black means that starch was present. And those spots, it matched the original green areas of the leaf. So what we can say is that only areas with chlorophyll were able to do photosynthesis and store starch. So the next thing that we did was paper chromatography. So paper chromatography, it's going to separate different plant pigments. This technique, it separates molecules based on their size, solubility, and polarity. So separation due to size, due to solubility, and to polarity. So the key thing to understand is that the lighter, so the less heavy or the more soluble a pigment, the farther up it's going to move up that paper. But a heavier pigment, it's going to stay closer to that origin. So what we did was we were able to identify carotenes or carotenoids as dark yellow. So they actually moved up the farthest. We were able to identify xanthophylls as light yellow, chlorophyll A as dark green, and then chlorophyll B as light green. And finally, we used a spectrophotometer to measure absorbance of these pigments at different wavelengths. So when we're talking about our dependent and independent variables, our dependent variable is going to be absorbance. Remember, our dependent variable is always going to be the y-axis. So that means that wavelength was our independent variable. The absorption spectrum we generated, it told us how much light each pigment absorbs across the visible spectrum. And that helps us understand which wavelengths are most useful for photosynthesis. So what we found was that chlorophyll A, it best absorbed around 425 nanometers. And 675 nanometers. So let's do for chlorophyll A, we're gonna do pink. So here we have around 425 and again at 675. We found that chlorophyll B, we're gonna do brown, it best absorbs around 450 to 475 nanometers. We found that our xanthophylls that we're gonna make orange, they best absorbed around 425 to 450 nanometers. And then we found out that our carotenes, they best absorbed around 450 nanometers. So these peaks, they show us that the plants, that they are using mostly blue and red light for photosynthesis. So I just did want to clarify exactly how absorbance relates to color. A pigment appears a certain color because it reflects or transmits that color's wavelength, and then it absorbs the rest. So chlorophyll, it looks green because it does not absorb green light. It reflects it. It's actually absorbing violet, blue, and red. Our carotenes and xanthophylls, they look yellow or orange because they reflect those colors and then absorb blue/violet. So that's all for Lab 7. I answered all of the study guide questions, but it is important that you guys, you need to know more than just this. This is just what was on the study guide. In the next video, we're going to go over our Lab 8. So I'll see you then.